Great. Well, thank you very much for the invitation, Anthony. What I'll be talking about is a molecule called monoamine oxidase A and its relationship in different imaging modalities in antisocial personality disorder. So we'll talk about the enzyme and the genetics, MAO-A gene variants and ASPD, a PET study of ASPD, and then genetic, structural, and functional MRI studies in antisocial PD. Okay, so monoamine oxidase A, or MAO-A. So MAO-A is a mitochondrial enzyme that's predominantly found in cholinergic neurons in the brain, and the highest concentration is found in the locus coeruleus. So it has a number of functions. One, it breaks down amine neurotransmitters like serotonin, norepinephrine, dopamine. It increases cellular oxidative stress levels, facilitates induction of apoptotic pathways. Those are cell death pathways, and it's a key molecule in mammalian neurodevelopment. It's an X-linked gene, and a variable nucleotide tandem repeat has been found 1.2 kilobases upstream of the transcription start site. So multiple copies of this VNTR exist, and alleles with 3.5 or 4 copies are transcribed 2 to 10 times more efficiently than the ones with 2 or 3 repeats. So we dub these MAO-A high or MAO-A low. Okay, so as most of you know, ASPD is a chronic psychiatric condition that first onsets during youth as conduct disorder. There's a pervasive pattern of disregard for and the violation of rights of others occurring since age 15. And then they list the number of symptoms there that are found in the DSM. And it's importantly for all of our studies, we used the SCID-2, which was based on DSM-4. Not sure how many of you would know about the Dunedin Multidisciplinary Health and Development Study. So this took place in New Zealand, and it's followed the lives of 1,037 babies born between April 1st, 1972 and March 31st, 1973. And they were all born in a single hospital in Dunedin, New Zealand. And they've been assessed at various time points in their life, so from age 3 to 38. And they get interviews, physical exams, blood work, etc., and they fill out questionnaires. And there's an extremely high retention rate because let's say one of these people moved to the States. They'll actually fly you back to New Zealand in order to get your data and not lose a data point. So there's a very low attrition rate. Now, a seminal study found that the interaction of the low activity MAO-A gene and severe maltreatment from ages 3 to 11 was associated with conduct disorder, disposition towards violence, conviction for violence offenses, and for ASPD symptoms. I'll turn to our PET study now. So why would we study MAO-A in antisocial PD? Well, 2 PET studies have found inverse relationships between self-report trait measures of regression and impulsivity, with decreased binding in orbital frontal cortex in healthy populations. And one of these studies was from Nellie Elliott Klein from Mount Sinai. There's a mutation of the MAO-A gene that results in complete and selective deficiency of the enzyme, and it's associated with very aggressive and impulsive behaviors. So in a Dutch pedigree, all the males who had this, this mutation, they were all convicted for things like exhibitionism, attempted rape, arsonism, attempted murder. There's been 2 mice knockout models that result in aggressive and impulsive phenotypes. And I already mentioned that the low activity allele in combination with abuse leads to more antisocial outcomes later on. So PET is a nuclear medicine functional imaging technique that is used to discern metabolic processes in the brain or body. The system detects pairs of gamma rays emitted indirectly by the positron emission radioisotope, the tracer, which is introduced into the body in a biologically active molecule. So the tracer that we used was harmine for MAOA. It has very desirable characteristics for binding to MAOA that are listed here. So we scanned 18 subjects with ASPD, 9 of which had comorbid alcohol dependence. And then our control groups were 9 male healthy subjects and 9 subjects with alcohol dependence. And so the inclusion criteria, the ASPD had SCID2 diagnosis, the healthy had no history of psych illness, and the alcohol dependent had no other psych illness, and no one was on any medications. Exclusion criteria were a history of a mood or psychotic disorder, current substance dependence, and cigarette smoking. And the reason we have to exclude cigarette smoking is that it actually inhibits the MAO enzyme, so that's why it's important to exclude smokers. So what we found here, so each white triangle is an ASPD participant, and each black one is a control participant. So we found in our 2 hypothesized regions of interest, the orbital frontal cortex and the ventral striatum, that MAOA distribution volume was lower in these 2 regions. And you actually see a global effect, a lower MAOA binding across the regions sampled. And this is not surprising because there's very high correlations between the different regions. When we looked at the relationship between MAOA and impulsivity in the ventral striatum, we found a negative correlation, such that those who were more impulsive had lower amounts of the enzyme. This was also true of psychopathic traits and scores on the Iowa gambling task. So in conclusion, this is the first study to demonstrate a reduction of MAO in regions that influence aggression and impulsivity in a pathological sample. Rodent model suggests that decreased MAOA during neurodevelopment result in excessive serotonin levels that adversely interfere with the CNS development and predispose to pathological aggressive and impulsivity. So our study supports an extension of this model into pathological human aggression and impulsivity. And then the reduction of MAOA in the ventral striatum suggests a mechanism to account for increased dopamine release in people with elevated psychopathic traits and impulsivity, given the role of MAOA in metabolizing dopamine. So now we'll move on to the MRI studies. So I mentioned impulsivity is a core feature of ASPD, and the ventral striatum is a region of neural circuitry underlying impulsive behavior. But the relationship between phenotypic brain markers, so MAO enzyme, and brain function had been unknown. So we investigated the relationship between MAOA distribution volume in the ventral striatum and resting state fMRI in bilateral inferior and superior ventral striatal seeds. And this was just in the ASPD group only. So we found patterns of connectivity between the ventral striatum superior and bilateral dorsal medial prefrontal cortex, bilateral pre and post central gyrus, and the right superior frontal gyrus that correlated with MAOA distribution volume. And we also found that the inferior ventral striatum seed predicted patterns of activity in limbic regions and posterior cortical regions that were anti-correlated with MAOA. And what you see in the red are negative correlations between different functional connectivities and markers of impulsivity, showing that the functional strength was inversely related to markers of impulsivity in this population. So takeaway, MAOA may be a neuromodulatory influence on neural circuits underlying impulsivity. The correlation of ventral striatum MAOA distribution volume with the ventral striatum superior dorsal medial prefrontal cortex functional connectivity suggests that activation of corticostriatal pathways implicated in impulsivity may relate to level of MAOA in the ventral striatum. And weakened striatal hippocampal connections in ASPD relate to ventral striatum MAOA level and may comprise neural substrates of impulsivity in this population. Next, we looked at a structural study of MAOA polymorphisms in ASPD, and this was structural MRI. We chose the amygdala because it's involved in moral reasoning, stimulus reinforcement, learning, and harm avoidance, as well as emotion processing. And it's a key region or an area of interest in ASPD. So 2 structural findings report abnormal amygdala morphology in ASPD. Another study of healthy subjects found reduced amygdala volumes in the low activity carriers relative to the high activity carriers. So we hypothesized a gene diagnosis interaction, and we had 18 ASPD and 20 healthy controls. So what we found was a main effective genotype. So those with the high activity allele, they had larger amygdala volumes than the low activity allele. And we also found a group by diagnosis or group by genotype interaction present in the right amygdala, such that the ASPD group with the low activity allele had reduced surface area in the right basolateral nucleus and increased area in the right anterior cortical amygdaloid nucleus. And then again, among the low activity carriers, the right basolateral nucleus was or showed an inverse relationship with polypsychopathic traits, such that the higher psychopathic scores were associated with the smaller surface areas. So in conclusion, acquisition of fear learning depends on intact basolateral nucleus, and serotonin is a neuromodulator of BLA neurons, so it's conceivable that the effects of MAOA on the system adversely affect the serotonergic functioning and the deficits that accompany it. The primary function of the anterior amygdaloid centrifugal nucleus is to process smell from olfactory bulbs, and psychopathy, interestingly, is associated with less efficient sense of smell. So our last study was looking at resting state fMRI and MAOA genetic variants in ASPD. So one study of healthy humans examined resting state fMRI in relation to the genetic variants, and they found decreased right middle frontal gyrus activity in MAOA-L and increased dorsal activity in the high activity MAOA allele. What we did was a seed-based correlational analysis from a priori seed regions in the ventral striatum and the dorsal caudate. And we had 19 ASPD and 21 healthy controls. Okay, so there was a main effect of genotype in the superior ventral striatum, and this depicts significant clusters of functional connectivity to the superior ventral striatum that differed between high and low genotypes. There was greater functional connectivity in the operculum, insula, inferior parietal lobule, and the occipital cortex in the low activity MAOA. And higher functional connectivity was found in the right frontal pole. This depicts dorsal striatal functional connectivity that showed a genotype by diagnosis interaction. And what we're seeing here are post hoc mean parameter estimates of dorsal caudate functional connectivity of the ACC, the frontal pole, and the paracingulate cortex. And the largest effects in ASPD were found with the high activity MAO allele. Increased functional connectivity between the dorsal caudate and temporal lobe, OFC, amygdala, and hippocampus, which were correlated with proactive aggression. And this was driven mainly by the ASPD MAOA low group. So discussion points. First study to examine association of MAO polymorphism with resting state fMRI in a clinically severe group. In healthy populations undergoing MRI tasks, typically used in aggression, we see increased activation of the OFC and ACC, as was seen in the high-activity MAOA alleles. And we suggest that increased functional connectivity of the dorsal caudate to frontal structures could relate to the neural basis of aggression in ASPD with high-activity MAOA. And the association of proactive aggression with caudate functional connectivity to limbic structures agrees with previous research showing activation of similar structures during aggression-based tasks in highly psychopathic populations. Okay, so MAOA-L polymorphisms with or without childhood maltreatment are related to violence and aggression, and which are key symptoms in ASPD. The PET data suggests that MAOA enzyme density is lower in OFC and the ventral striatum of ASPD. And ventral striatum resting state functional connectivity in ASPD may relate to ventral striatal MAOA enzyme level or genetic variants. And amygdala morphology may also relate to MAOA genetic variants in ASPD. So with that, I want to thank my lab members, funding sources, and my collaborators. So thanks very much.